I think I will start the meeting because participant is enough for the meeting. So, hi everyone, happy to see all of you, and welcome back to today's session organized by VODAN Africa, and my name is Putu Hadi. So we have some agendas today. Let me share my screen. So the first agenda is start from the opening. So maybe I will hand over to Dr. Ibrahim, and then the second agenda, Katie will present about digital health and science research projects at Leiden University of Advanced Computer Science and LUMC. And next agenda is country coordinator update. Maybe Dr. Reginald and I will share about that. And Prof. Francisco. Okay. And then we can move to the next agenda. It is finance. If Frederick is here, and any other business that if someone wants to give a feedback or a comment. But now, well, I see Prof. Francisca is here. Maybe I will give the floor to Prof. Francisca to lead the meeting. So Prof. Francisca, oh sorry. So during the meeting, so please mute your audio, and please raise your hand or comment on the chat box while the presentation is going on. And now I will give the floor to Professor Francisca to lead the meeting. Professor Francisca, the floor is yours.
Thank you very much, Putu, and it's a pleasure to have every one of us here today for the weekly VODAN Africa meeting of Tuesdays. To thank you all for keeping faith with the network and for everything you have done to ensure that we achieve our milestones. I have been in touch with the technical team, and I can say that we are actually in time to the deployment for this month. And I can also report that the funding for the computers have been disbursed to the different countries, and the computers are being sourced for as we speak. So we're still also in time for the overall deployment in addition to the pilots. I would like to especially welcome Katie for accepting to speak at today's event and today's meeting. And I can assure you that we're going to learn so much from her, from the work she has been doing in Leiden with the digital health and the fair health data management, which interfaces excellently with what we're doing in VODAN Africa. To also thank you, Putu, Aliyah, and Mariam for setting up this meeting and for fine-tuning the agenda. As agreed, Dr. Ibrahim will be chairing today's meeting. Professor Miriam is unavailable, even though I'm available, but we feel it is good to give the country coordinators some coordinating quote role now in the meeting. So I'm going to probably be stepping aside while I listen as everything is going, while Dr. Ibrahim Abdullahi, the Nigeria co-coordinator, leads the rest of the meeting. So Putu, I'm trying to see the agenda now, the final agenda that has been drafted. Can I see that?
Yes, in the script, it's also in the chat box, Prof.
Yes, I got that now in the opening, yes. So I have the pleasure and honor to invite Katie. If you wish to share your screen, Katie, you should do that now to speak on the digital health research, digital health and science research projects at Leiden University Institute of Advanced Computer Science, LIACS. I would like to that, I'm sorry, Katie, that Katie is one of the supervisors in GAIC. You remember GAIC is the research arm of VODAN Africa, where we have most of our PhD students and professors as supervisors. Yes, Katie, apologies for interrupting you. You can have the floor now.
No problem at all. Thank you very much, Professor Francisca, for the kind invitation to speak today. Miriam asked me to talk a bit about all of the different fair data applications I've been working on in Leiden and how they might relate to VODAN. Of course, there's a lot of overlap. And so that's what I want to focus on today. I am based at the Computer Science Institute in Leiden, but my my real background is in biology. So I started as a biochemist, and I've been working in bioinformatics for the rest of my research career. I work quite heavily on the computer science side of bioinformatics these days, but at heart, I'm still a biologist. So a lot of the projects I'm going to talk to you about today are about enabling various different biological, biomedical communities to share and exploit their data in fairer ways. And so I want to focus on a number of applications and approaches that I've been looking at recently. And whenever I think about fair data, I like to think about, well, the purpose of the verification, why we are verifying data in the first place, who we are verifying it for, and for what purpose. And when you think about it like that, then there are various different places in the research life cycle, in the data life cycle, that verification has to take place. And so the first two applications I'm going to talk to you about involve what I like to call fair by design or fair by increment. And the other two that I'll talk about after that are about verifying existing data. And I was trying to think where VODAN fits into this scheme. And actually, I think it's somewhere in the middle. And I want to explain a little bit more about these different concepts before I come back to that point and to explain why I say that. So what I mean by fair by design is that in the life sciences, in molecular biology in particular, we can generate a lot of data very quickly now with all sorts of new omics technologies. Now, for some of those technologies, we can generate data automatically in standardized formats with standardized annotation. And so from the beginning, we can generate data which is in some way fair. Now, that doesn't include information about the context of the data, the experiments that produce the data, what kinds of extra functional annotation we might want to add to individual molecules in that data. And so fair by design is a good place to start, but actually generally what you want is a platform that will allow you to ingest automatically, ingest fair data automatically, and then augment it as you go along. So this is what we mean by fair by increment. And then finally, verifying existing data. Well, a lot of the time we have large data sets, but they're not in any kind of fair format. They're very difficult to integrate with other resources. They're very difficult to exploit to their fullest. And so you want to be able to access that legacy data and make it more useful, make it more interoperable with other data sets. And so I'm going to start with the fair by design, fair by increment. So these two projects that I mentioned, one is in systems biology and the other is in light microscopy. The VODAN project I've been involved with for the longest. We started back in 2008, so it's a very old project for computer science for academia. And in systems biology, we have a lot of heterogeneous data. We link data to mathematical models and to bioinformatics processes. The people who work in systems biology projects tend to be distributed. So there'll be lots of people working on the same project, but in various different locations. And so what we wanted to do there was produce a platform that allowed all of this heterogeneity to take place and allowed people to come together to make verifying and sharing easier. And the result is an EU platform for collaboration and exchange. So this is a platform that supports a running consortium doing their experiments, producing their research results, but it also, when it's time to publish the data, becomes a dissemination platform. So before publication, you can keep all of your data private to your own project or to individuals that you name. But when it's time to publish, then you can disseminate your research results through the platform. For the other project that I mentioned, the NL Bioimaging project, we are supporting the light microscopy community there. Again, we have heterogeneous data, but this time heterogeneous imaging types. We have to link those images with all sorts of different types of processes to analyze the image data. Unlike the systems biology project, we have people generating data and managing it in a much more local way, because quite often this data is enormous and you don't want to be transferring it across the network. But after you have done the processing, you want to be able to interlink your results with other people in other places in a national network or a European global network, depending on the project. So if we just have a brief look at some of the components of these projects under the hood. This is a screenshot of the VODAN hub, which is the systems biology platform that I mentioned. And as you can see, we deal with the heterogeneity of the data by providing a framework to describe how different experiments fit together. And we use a common standard that's already available in the community called ISA, Investigation, Studies and Assays. And the ISA framework, we extended a little bit to enable us to talk about the mathematical modeling components of systems biology. And so now we can say this data was used to generate this model, this data was used to validate and test this model. All of this data comes from the same study, which comes from the same investigation. These are the samples, the biological samples that were used to create, to generate the data in this study. These were the models, these were the standard operating procedures, processes and protocols. And so we have this integrated view of this heterogeneous, multi-faceted experiment involving multiple different types of scientists, based on all sorts of community standards. But those standards, individual scientists don't need to know them all. They just need to follow our recommendations. And under the hood, we do that by sharing templates for various different types of data. Now, each different type of data has a different set of metadata that it requires, but there is overlap between these metadata schemes. Where there is overlap, we can aggregate data. Where there is no overlap, it doesn't matter, we don't have to worry about that. And this is because we follow the linked data paradigm. So we generate automatically RDF linked data from our template tools that we developed as part of this project. And the main tool that we use is something called Writefield. So that allows us to embed ontology terms into just regular spreadsheets and then automatically extract those ontology terms and the metadata in RDF through the same mechanism. And so we hide away a lot of the complexity of annotation from our users. At the same time, we provide, whoops, that's the wrong way with the slides, a lot of tooling that fits into the platform to allow our users to make the best use of the data once it's in these FAIR formats. So we provide incentives for joining in with what we recommend. Now, for the second project, the NL Bioimaging project, this came later. And what I really wanted to do was apply everything I learned from the VODAN project in systems biology to this community. And one of the main things that we learned was that we have to build on top of resources and tools and standards that already exist. The light microscopy community was quite a way behind the bioinformatics and systems biology community in terms of metadata standards. But what they did have was something called the open microscopy environment set of standards, which allow common representations of images from various different microscopes. So every microscope has their own proprietary format. The OME standards allow those proprietary formats to be exchanged. And so that means it doesn't matter what kind of microscope you use, you can share the images with the OME standards. And there's a database called Omiro that goes along with those standards. And so everything that we did in NL bioimaging built on top of the Omiro database. But what you can say in terms of the metadata in Omiro wasn't very well specified. So what we've been doing in the last year and a half, is verifying Omiro, if you like. So we've been developing metadata templates in a similar way that we did in the systems biology project. We've been using the same components, the right field spreadsheet tool that I told you about. And we've been embedding these templates in emerging standards, so something called REMBI, which is the recommended metadata for bioimaging experiments. And it's just come out recently. And so we are helping this user community start to understand, well, the value of having fair data, but also start to understand new processes for generating and annotating data in a fair way. And we're providing tooling to make the barrier of adoption of that as low as possible. Now, along with the metadata that we need to generate at the beginning of the experiment, we need to generate information and metadata about what happens during the experiment. So as data is analyzed and processed, we need to be able to add extra metadata and results back to the same experiments. And so we've been working as well on a set of methods for producing fair workflows. So common ways of accessing, augmenting, and interlinking data, and sharing the actual analysis protocols and methods that people use. And so we've taken something that already exists and we've made it into more of a fair resource. That work is still ongoing. We've also interlinked Omiro with the local electronic lab notebooks so that scientists don't have to enter their data twice. So simple things like this really help with adoption of these kinds of tools. Now, what we've been doing in Leiden with this Omiro database is a pilot for a national rollout. And that's what we're doing in the NL bioimaging project. We have this example project in Leiden. We are now in the process of rolling this out to another 4 nodes in the national network in this ring you can see here. And we're developing a national node, which will not store data centrally, but it will just become a kind of registry of metadata to allow people from different nodes to understand what each other is doing and to foster communication. Now, when data is published from any of the individual nodes, that's when it needs to be shared externally. And there are European resources already, the image data resource and the bioimage archive that will allow that publishing. So we're providing automated mechanisms for getting data from these local resources into these public resources. And the image data repository is also based on Omiro. So in terms of augmenting Omiro locally, we are hoping that we can also help to make the central image data repository fairer as well. So both of these projects are really doing quite similar things. We're supporting scientists doing fundamental research in various different biological communities, and we're supporting them through the process of doing their experiments. So it's about supporting the collaboration, the development of projects as they go along. And for that, you have to build on what's already there. You have to fit in with common standards, common practices. You have to understand that if an individual scientist is going to spend time doing verification, there has to be some incentive for them. And you have to take these communities along with you. So incremental development is good. If people see some benefit, then they can start to use your tools. And as you develop your tools further, there's more and more benefit to come. And if there are requirements from funding agencies and the government and such things for compliance with various sharing and archiving procedures, then if your platforms provide those services, then that also helps with adoption and spreading the verification message. So these are about fair by design, fair by increment, but verifying existing data is in some ways more of a common thing to do. So we already have this massive legacy of data. We know that some of it is not being used to its full potential. We're missing important links between related data sets. Simple comparisons are not that easy. And if you want to reuse data, there's a big barrier to get over to enable that to happen, because it's not in a format that lends itself to any kind of integration. So verifying existing data is incredibly important as well. Now, I work at LIAC, the Computer Science Institute, but I also have a position at the LUMC with the biosemantics group there. And that group has pioneered a workflow for the verification of existing data. And this is presented in the FAIR Data Special Issue in Data Intelligence. And the paper that I'm referring to mainly here is by Anneke Jacobson and colleagues. And here she presents the way that verifying of existing data generally goes. So first of all, it's really important to understand why you're verifying the data, who you're verifying it for, what your main objective is. Then you need to analyze what data and metadata you have available, and also how that data and metadata fits in with any existing community standards, whether there are any community standards. If there are not community standards, can you define semantic data and metadata models for yourself? Once you get to that point, then you can start to actually make the data and the metadata linkable. And it's at that point then that you can actually host FAIR data. Now, it could be that you only want to host the metadata. It could be that you want to host the data and the metadata. That really depends on the purpose of the verification and on the type of data that you have. A lot of the work that the biosemantics group does is with rare diseases. So rare disease registries, various rare disease communities. And so it's privacy sensitive data. And a lot of the time when you talk to people who own privacy sensitive data, then there's some reticence about sharing. There's a belief that you can't share because it should stay private. Now, of course, FAIR data, we all know, is not the same as open data. The accessibility part of FAIR data is incredibly important. It's perfectly possible to have fair, privacy sensitive data, and there are lots of advantages for making fair, privacy sensitive data. And so with the final part of this talk, I want to talk about a couple of other projects which sit in this space. So these are existing data sets that we verified using Anneke's workflow. And these are privacy sensitive data sets. So they involve patient data, but the purposes of verification in each of these cases is quite different. So the benefit project is about developing and monitoring interventions to promote healthy lifestyle and modify risk in cardiovascular disease. So they've got lots of cardiovascular disease patients who've had cardiovascular disease episodes, and they have been suggesting modifications to lifestyle, and then they've been monitoring these patients and observing all sorts of changes to their health over time. And so this data set is now a very rich data set that will enable new types of research into prevention approaches in cardiovascular disease. And so they want to be able to share that more broadly. The other project I want to mention is something called HEBON, which is the Hereditary Breast and Ovarian Cancer Research Network in the Netherlands. And this is a nationwide survey of families where breast and ovarian cancer is common. And it's been collected for about 25 years. So it's a very big data set that's been collected over a long period of time. And there, it's not about sharing the data more broadly, it's about making sure the data that they do have is up to date, because things change very rapidly in cancer research. And I'll go on to talk about that in a minute, but this is what the HEBON data currently looks like. We're in the process of verifying that. The project that I was working on is actually a pilot implementation to show the value of doing this approach. So the HEBON database is actually a distributed database. Different types of data are stored at various different institutes across the Netherlands. And when people come to ask a research question, of course, there is an ethics committee that decides whether they should be allowed to ask this research question or not. But once that ethics committee has approved some research, then somebody from HEBON has to integrate the data sets required to answer those questions. And this is quite costly in terms of time. And so, of course, if they had fair data, it would be very easy to generate the data sets required for each individual request. And that's where they're moving to. So it's not about generating this data so that you can distribute it more broadly. It's still going to be very well controlled. But it's going to save time for the people in the HEBON project in terms of generating the data. And more importantly, it's going to mean that the data that they give to people is the most up-to-date and most current. Because if you look at just the genetic data in the HEBON database, the DNA data, 25 years ago, we didn't know quite as much as we do now about various different pathogenic variants which are related to breast cancer. And so this picture shows BRCA1 and BRCA2, the most famous genes associated with breast cancer. They are not the only ones. Over the years, many other genes have been added to the list, and variants within those genes have been causally linked to breast cancer. But every time a new variant is discovered, the people in HEBON have to go back to their data set and identify whether there are new variants. These new variants actually occur in any of the data. And so it's not about making the HEBON data more available, it's about making the public data more available to people within HEBON so that they can reanalyze the data that they have. And of course, in recent years, we've started to learn a lot more about combinations of variants and the risk factors involved in having combinations of those variants. And so following Annika's workflow, what we did in the pilot project was we analyzed the data that we had. We looked at the community standards, community ontologies that were available. We annotated, re-annotated, and represented the data in these standard formats. And by doing that, it meant that we could then interlink the HEBON data much more easily with public resources. Now, we're not sending the data out, we're getting the public resource data in to use with the HEBON data. And so this is the result. We can now query all of the major public resources very easily for the latest information about variants and integrate that very easily with the HEBON DNA data. Every time a new analysis is run, we can keep track of the results and the provenance of that. So the data is not available for querying, it's still controlled, but it's much easier for researchers to query the context of that data. In terms of the benefit project, what we have is in some ways a similar situation. So we have various different types of data spread through a number of different locations, and we want to be able to pull those together. And by pulling those together, it enables a new set of research questions to be answered. And these are just some example research questions that some of the researchers within Benefit came up with. But the process of verifying this data, the legacy data, is very similar. So these are the types of data we have. There are emerging community standards that we can use to represent these types of data. In this case, we are using something called OMOP and SNOMED codes, which I know you're familiar with in VODAN, to standardize the way that the data is represented. And we're using a common data elements semantic model, which was actually developed in the European project of joint project of rare diseases, so from the biosemantics group and others, to represent the relationships between the observations and the clinical findings and the procedures in this data set. So again, we're following the verification workflow. We've analyzed the data and the metadata. We've found common representations based on community standards for that. And then we've decided, in this case, which metadata we would like to expose to enable others to identify interesting data for their use. Now, again, it's privacy sensitive data, so actually we're not going to be exposing the data, but for people who are able and given permission to use it, then there is also in development a secure environment based on Andrea to do further analyses on this data. And so I hope I've given you a little bit of a flavor of the different types of verification and the different purposes of verification that I've encountered recently. And I hope now that you realize why I say VODAN sits in between fair by design and increment and verifying existing data, because we have a situation where there were already procedures for collecting data, quite often on paper, sometimes electronically, which have now been formalized and standardized using shared vocabularies and common queries using the CEDA framework. And there's a lot of focus on keeping data and the control of data local. And so the question there is, whereabouts is VODAN in the verification workflow? Are we all the way through that workflow? Are we halfway through that workflow? Which steps are now the bottlenecks? But then on the other side, fair by design and fair by implementation, what you have now is a system for future data collection, which is already in a fair format. The benefit of collaboration and sharing is becoming apparent to the network of involved institutes. And the question there then is, what's the end goal for the technological infrastructure? How far through the linked data semantic web world will VODAN go? We have a mixture at the moment of relational data, CSV data, we have semantic annotation of data. And so what's the end goal there in terms of representing all of the data? So I think it's time really to draw my presentation to a close. What we have is, if you're going to do any verification, it's very important to have clear verification goals. You have to know why you're doing this process and to be able to incentivize the people involved to join in. You have to make it as easy as possible. Whoever is going to join in, whether they are researchers, clinicians, PIs, whole institutes, it has to be a straightforward process. But now that we have a lot of fair data projects out there, the added value of fair data is a large incentive in and of itself. So I think sharing experiences and success stories like this is actually very important for generating and building capacity in the future. And with that, I would like to stop and thank all of my colleagues from various different infrastructures and developments that I've talked about, and ask if anybody has questions.
Wow, what a wonderful presentation by Katie. And we really want to appreciate you. Don't mind me, I just jumped in to speak before Prof. Francisca. It just demonstrates how involved you have taken all of us around, and it justifies and also make the point of stamp that you are an expert in fair data. We are so proud to have you. And lest I forget, after this presentation, I know many of us will want to have your email so that we can have a proper collaboration with you. Already from here, I can see information-centric networking coming into fair data science, and I will want to share a few points with you so that we can see what we can do moving ahead. So, Prof. Francisca, for your comments, please. All right, if Prof. Francisca is not speaking yet, maybe we have questions. Let's just take a few questions before we move on to the next point. Yeah, we have questions from our team. Any team?
I have a question.
Hello? Okay.
Okay, yeah, please go ahead.
So I saw, I want to talk about verifying the data sets that already exist. So,
This after?
yeah, that's me.
All right, please go ahead.
So with the data sets that already exist, when you're verifying them, especially for medical data, then I assume you're going to use something like ICD or SNOMED or LOINC or whatever. I think you said that you're using SNOMED and OMOP for this particular exercise. Yeah. So who annotates? Do you annotate it at the clinical level, or do you annotate it way later? Who annotates this data?
Well, actually, some of it was already annotated with SNOMED codes. So that made life very easy in this particular project. And the only real thing that we had to do was map the SNOMED codes to OMOP terms. But yeah, in this case, from the beginning of the project, the researchers involved knew that they wanted to share the data and they wanted to make sure that that was future-proof. And so SNOMED was used from the beginning. If you don't have any kind of annotation, then the only real way to do this is for clinicians and data people to sit down together and do this mapping process together.
Okay, so has there been such a situation where you did not have any annotated data?
Yes, yeah, in other projects we've had that situation, and at the beginning of the systems biology project, we had a few example data sets that needed to be produced in the common standards for the community. Now, these were not medical data, it was bacteria mostly at the time. So the process was quite straightforward because I was allowed to see all of the data, and I just simply sat down next to somebody in their lab and went through the whole process with them. But of course, that's not necessarily that scalable. So the earlier you can do this kind of annotation, and the earlier you can provide templates and ways of working to allow the scientists to do that for themselves afterwards, the better, actually.
All right, thank you.
Yeah, please, Ablaikao, you want to make a brief question also?
Yeah, I think mine is in contrast to it, Tia, like, so when the data is does not exist, so how is the workflow again?
When the data does not exist. So you're not talking about verifying existing data, you're talking about the fair by design, fair by increment.
Okay.
Right?
Yeah, so my point
Yeah.
is this, like, so in case of the benefit project, I want to think maybe the data may not, let's say we don't have existing data, right? So in terms of the workflow, what are some proactive activities that went into that?
So if you're trying to do the verification process before any data has been generated, you're in a very strong position because you can ensure that the data that you do generate is as standardized as possible. So there you're just looking at the community recommendations, community standards, community ontologies and vocabularies available to you. And then you just need to instantiate those in an easy way for people to use. Now, in our projects, that's right field. In VODAN, this is CDAR. So these are metadata management frameworks, if you like. That's the important thing there.
Yeah, thank you.
All right. Thank you so much, Katie. Sure, if we want to go with the presentation, we will not end the questions. And very important for us to keep you busy and also to keep you engaged in your expertise as a fair data expert. So moving ahead, Putu, I think we'll take a few updates from Dr. Regina and Putu yourself. And then after that, we will take some reports from Alia also. Then Frederick, Oluwole, and the Mariam Basaja team. Thank you.
Okay, thanks, Katie, for your excellent presentation, and also Dr. Ibrahim. And I think I will, yeah.
I think
Mariam
has a question.
Oh, Samson still. Oh yeah, sorry. Samson, do you want to? Yeah, you can unmute yourself.
I think if I may, I'll try to make it short, I think, because of the time. It was a very insightful presentation, Katie. You showed us the application areas and extent of how FAIR has gone so far, and it's really inspiring. When I come to my question, in relation to Omero, it says it keeps your data on central location. And is it against the FAIR principle? I mean, or are we saying the central location to be your central location? I mean, in the local central kind of thing. How does it relate with a federated concept? And the templates that you create, which tools are you using, and the tools that you also use, are they proprietary or open source? Looks like too many questions. Over to you.
Yeah, that's okay. So I'll start with the first question. So Omero is more of a traditional database, if you like. So it's designed for data storage. And so if you instantiate an Omero instance, then it really is a database where people put their data centrally. Now, the way that we've set it up in NL Bioimaging is that each node has their own Omero, and then we have a national Omero for sharing and collaboration. So this is not against the FAIR principles. There's nothing in the FAIR principles that says you can't have a central database. What you should have is a way of interlinking what's in that database to other databases. And that's what the national node is for, and that's what the European IDR is for. For your second question, the metadata templates we developed, actually we just use Excel wherever possible, because that is the standard format that most people in laboratories really actually use. But we've augmented Excel with a tool called Writefield, and that's open source. So we have a general policy, both in my previous lab in Manchester and also in Leiden, to be as open as possible with software. So yeah, if we've developed it, then it's going to be open source and available. And Omero is also open source. It's something that is just available for the community.
Thanks, Katie.
Okay, is there any question before I continue the agenda? Esther, do you want to ask? Yeah.
I'm okay, thank you.
Okay.
Oh, my hand is still up, didn't notice.
Yeah. Okay, then if there is no question anymore, so I will move to the next agenda. Thanks, Kathy, for your presentation, and I will move to the next agenda. It is update from country coordinator, but before Dr. Reginald will continue my update, I will make a short summary about what we discussed yesterday. Basically, yesterday we have a fruitful discussion with country coordinator. It is about the VODAN agreement with health facilities, but some adaptive change should be required to make with and now I'm still working on it. So at least tomorrow I will share it to you, because I need to recheck the video and like make a notes about what point, what is the important point, and will adapt the agreement to the result of the discussion. So at least tomorrow before 1 p.m. CET, I will share it to the country coordinator, and country coordinator could print the agreement and maybe make some notes about that, and we can have a discussion about that next month in the Monday's meeting. So that's all from me, and Dr. Reginald, you can continue the update from the country coordinator.
I think what you have covered most of the discussions. I think the question here is the agreement. If the agreement is taken to a health facility, who should sign? Is it the data controller? Is it the CEO of the hospital? Is it the security officer, data clerk, data requester? You find that some of these processes can be covered by one person or two people. So this is what we are discussing to understand if the agreement is signed at the health facility level, who should sign? Who are the key people? And then the country coordinator, and then perhaps the endorsement of the Minister of Health. Some health facilities are already government facilities, so may not require the Minister of Health because they represent the Minister of Health. And then the last discussion was, so what about the role of VODAN? What is VODAN's role here? So there's a question about dynamic data controller, and where would that person be? In Leiden or somewhere in outer space? Or should we create it at Kampala International University? It's a big question. And then at the country level, who is really the person responsible for this data? So those are the key questions that we're trying to discuss. And thank you, Putu, because you're trying to realign these processes according to each country's needs. Thank you.
Okay, thanks, Dr. Reginald. And I think I will give it back to Dr. Ibrahim, or maybe Mariam or Alia want to update the process of in VODAN Africa architecture. So Dr. Ibrahim or Mariam or Alia? Is Frederick here? So okay, then Frederick, do you want to update something from the finance?
Yeah, thank you, Putu. On my end, I think in the last meeting, I updated the team that we were basically going through the process of preparing all the resources needed for the purchase of the computers. And towards the end of last week, we accomplished that process, and we sent all the resources needed for the respective countries, that is Nigeria, Uganda, Kenya, and Tanzania. I do believe that some of them have received the monies, except one or two, which still have small challenges which we're trying to sort out. But then I guess from here now, I think the onus is on the country leads to proceed with the purchase of the computers and then just move on with the project, unless otherwise another financial need arises, which we will communicate from. That's basically it from my end.
Okay, thanks, Frederick, for your update. Well, there is a question from Esther. It is about fair equivalency publication, but I cannot answer it. I think Prof. Miriam is not, because Prof. Miriam is not here. Maybe Prof. Francisca, can you answer Esther question? Prof. Francisca? Oh, Prof. Francisca is not here. Well, Esther, I cannot answer it because I believe that Prof. Miriam is the best person to give you an answer about the progress of our publication. So then, Dr. Ibrahim, do you want to speak or state something before I close the meeting? Dr. Ibrahim? Okay, I think.
Hello.
Yeah, Getu, you can go ahead.
Maybe I have one question for. In fact, I would like to respond it to the first. So my question was, what effect does verifying the data in their project have in their analytics workflows of their projects? So perhaps Dr. Katie, maybe your response is important to the public too.
Hi, yeah, that's a nice question. I was just replying to that on the chat, actually. If you have fair data, then it should be easier to create and manage analytics workflows, because the context of those workflows is much clearer, and what is required for input, what is coming out of those workflows, the context there is much clearer as well. So it should mean that other researchers can interpret and criticize your analyses much more easily. And this is what we have seen in previous projects. I also did a lot of work in scientific workflows with a platform called My Experiment, which is now the Workflow Hub. The new version of this is the Workflow Hub. And that is about sharing workflows in the context of the experiments that they were designed to analyze. So yeah, fair data plays a really important role here.
Mm-hmm. Thank you very much.
Okay, before I close the meeting, is there any question or feedback?
No, Putu, sorry, please. Maybe we just take a very, very fast update from Frederick and is Ali on the call? And also if we have any new member for just a brief introduction before we close. Yeah, we know time is almost up.
Yeah, thanks, Dr. Ibrahim. Actually, Frederick already make a progress update, but I think Alia, do you want to update from your side?
Hi everyone, Putu, what exactly do you want me to update on the technical part, right?
Maybe you want to talk about the workbench or any little update you want to give to the house on Andrea?
So basically, yeah, I can add an update regarding the Andrea. I mean, like I've got the requirements for Andrea yesterday, and I'll work with those requirements with Eric and Lawrence from LUMC. Probably tomorrow we'll have a meeting. So yeah, that's basically it regarding Andrea.
Thank you so much, Aliyah. Good to hear from you also.
Yeah.
Hello, Dr. Ibrahim.
Yes, Getu.
Maybe a brief update from Magala team. We have completed the bulk upload. So anyone who wants to check it and try it out can find it in the VODAN GitHub repository. I have also shared it in the chat. Thank you.
Thank you so much, Getu, for this, and I hope many of us will try it out and see. Thank you. So over to you, Putu.
Okay, thanks, Dr. Ibrahim, and thanks, Alia and Getu for the update. I think well, it's already the time, so and I will close the meeting now. Thanks everyone for the participation of this meeting, and the recording of this meeting will be shared in YouTube and also in the WhatsApp group. So thanks once again for your participation, and I'll see you next week in the same meeting. Bye everyone.
Bye, Putu. Bye, everyone.
Bye-bye.